And in general, just thank you for the years of support and having me as a member of the school. Some of you I can call my friends. I'm very happy with that. I hope you will enjoy this talk. So I'll be talking about what I've been doing the past years during my PhD, trying to understand a little bit more about the evolutionary drivers of an adaptive radiation in Tillandsia. So a little outline of my talk. Will be giving a general introduction, explaining a little bit the terms and why it's interesting to study adaptive radiations. And then I'll go over 3 projects that I worked on in these years. The first project is more of a comparative genomics study. The second one goes more into functional questions with transcriptomics and also physiological measurements. And then the third project, we take a step back. We don't look as much into certain traits, but we just ask questions about the radiation of itself and certain potential drivers of it. And then just the general conclusions and acknowledgement section. So let's start with what an adaptive radiation is and why it is interesting to study them. So adaptive radiations are characterized as a burst of diversification in a specific lineage. So usually over short periods of time, you get from one common ancestor tons of different genetic lineages that arise. And in the case of an adaptive radiation, this is not just noticeable on the genetic level, let's say, but also on the ecological and phenotypic level. So we have a very wide range of ecological and phenotypic forms that arise, generally thought because the availability of new ecological niches is what is actually driving this diversification. And I always tend to talk about the cichlid radiation as an example. It's really a textbook example of adaptive radiations, where you can actually see that for the entire cichlid group that involves tons of non-radiating lineages, the 2 radiations of the great African lakes, Malawi and Victoria, take in a very significant proportion of the diversity in this group. And that goes together with very different shapes and forms and ecological adaptations. And in fact, adaptive radiations have been identified all over the tree of life and are really ubiquitous and a very important part of how biodiversity is generated. So that's one of the reasons why it's interesting to study, because they are part of how of speciation in general, but also because they are rather extreme cases where things happen very quickly, very suddenly. And sometimes looking at an extreme case can also give us valuable insights into general processes. Now, the main question that people have been trying to address in the last 50 years, and especially in the last, 20 years, maybe with the onset of sequencing, etc., is, if in such a short term we can have all these phenotypes and these ecological adaptations arise, where is all of that genomic variation actually coming from, that it's being recruited so differentially, so rapidly, right? And there are actually quite a few theories that arose. The more traditional, let's say, or an older theory is that we have innovations, certain traits evolve that are called key innovation traits that allow species to occupy novel niches, but that doesn't really address the question of the genomic variation. So other people have thought about, for example, gene flow having a very important role in creating hybrid lineages and combining already existing variation into novel combinations. There are other studies that point at the importance of what's called genomic potential, where you have just fast evolving genome evolution in certain lineages, and that also allows rapid radiation to occur. So in this talk, I'll try to look a bit into all of these different things and see if the Tillandsias are also characterized by any of those processes. So the subgenus Tillandsia, why do we study this subgenus? Well, first of all, a lot of radiation studies are concentrated in the animal kingdom, and it's definitely valuable to also have some plant counterparts. There are also other plant radiations that exist, but still, it's definitely one thing that makes it interesting. The subgenus Tillandsia is a group of about 350 species that belong to the Bromeliaceae family, which the most famous member is pineapple. Pineapple, however, is about 50 million years diverged from the subgenus Tillandsia, so they're not pineapples. And they belong to one of the fastest diversifying clades of the plant kingdom. So we recognize two lineages in the subgenus. We have the South American lineage and a Central and North American lineage, I should actually say. The South American lineage, there are about 50 species described. It has fewer species. The Central American lineage has about 300 described species. And it's thought that the origin of the subgenus Tillandsia is in the Andes, around the area where nowadays the South American species live and grow. And then 2 to 3 million years ago, according to previous studies, this Central American, North American group arose. So it's not just a clade that looks like it has diversified recently and fast. It's also characterized by several key innovation traits that I mentioned previously, are often characterized in adaptive radiations. And so these traits are theorized to actually allow lineages to occupy novel niches. And a few of them I will describe here. So quite a few Tillandsias have evolved the epiphytic lifestyle. So they don't grow on the soil, they grow on top of trees. And you can definitely see the competitive advantage of that. They escape competition from the undergrowth and have a higher position in the tree line of the forest, have more access to light, etc. However, it does also go hand in hand with losing access to a very important source of water, which would be obviously the soil. So often we also find other key innovation traits that have evolved as well in certain species and in others not. So for example, we have tank formation, where some of these plants, they have a very tight, like the inner leaves of the plant are very tightly wrapped so that they actually form kind of a bowl-like structure in which they can accumulate water and have water as an alternative source of water. You also have different layers of trichome density, or different degrees of trichome density in the radiation as well. Trichomes are leaf heads. They help plants absorb water, moisture from the atmosphere, and generally are an adaptation to aridity. And then very importantly, I'll talk quite a bit about this one. This is the evolution of Crustulacea and acid metabolism, short, let's call it just CAM in short. And this is an alternative mode of photosynthesis. I will describe more in detail exactly how it works later on in the talk. But what you have to remember of it is that it enhances the water use efficiency of a plant. And then there's also a very wide variety of flower color and bracts color as well. So you can kind of imagine how, with all these different key innovation traits, if they occur in different combinations, then definitely you're generating really diverse phenotypes, and they can occupy all kinds of different niches. And that's often sort of how this radiation has been described in the past. But what we don't understand exactly is how is this linked at all with genomic variation. So that's the first study, a little bit what we tried to get into. So we were trying to understand what kind of genomic variation is underlying, in this case, one specific key innovation trait, because it's a lot of work to focus on several at the same time. So we decided to focus just on one, and that is CAM, photosynthesis. So the idea here was to look into the genomes of the plants that belong to this radiation, describe them, compare them, but also try to understand if we find any association between certain types of variation and this specific trait. So what we did was to select two members of the radiation, Tillandsia fasciculata and Leiboldiana, that kind of represent the more extreme ends of the phenotypic range within the radiation. When it comes to CAM photosynthesis. So you could say that Tillandsia leiboldiana is more C3-like and Tillandsia fasciculata is more CAM-like. And then we went on and constructed reference genomes for both species. So I'll just briefly describe them because it's an important resource for the Bromeliaceae community. So we constructed reference genomes using PacBio, Hi-C data, and Illumina data in combination. And we obtained relatively contiguous assemblies. So the kind of base chromosome number or the chromosomal number that we're expecting in Bromeliaceae in general, it tends to not vary much, and it's about 25 haploid chromosomes. And we indeed recover 25 and a tiny 26th one for Labeldiana. And from that point onwards, the scaffolds become quite small. So we were quite happy to see that. And in terms of the gene content, you can also see that the orthologous genes are mostly grouping in these main scaffolds as well. So that was nice, because then we can also look into larger differences, such as rearrangements. We also measured the genome size of these two plants, so they vary. Okay, this cursor is interesting. Right, they vary a tiny bit. So Labeldiana is about 20% larger than Fasciculata. And that's mostly because of repetitive content, differential accumulation of repetitive content in the two plants. And so then we also wanted to look on the broad scale how they differ, starting with karyotypes. So we obtained the karyotypes of the two species, and we discovered, quite to our surprise, that we actually have a difference of 6 chromosome pairs between the two species, with Tillandsia Labeldiana having to apparently have 6 fewer than Tillandsia fasciculata and the majority of other Bromeliaceae. And then we did this synteny analysis using one-on-one orthologous genes between pineapple here and then fasciculata and Labeldiana. And you can definitely see that there is some rearrangements going on between Labeldiana and the two other plants. And we also recover specifically one fused scaffold and also 2 translocations. But unfortunately, it turned out that our reference genome was actually not that contiguous for Labeldiana, because we have 26 large scaffolds and not 19, as we would have wanted to. So we are a little bit limited in how much we can really study these rearrangements. But one thing that I wanted to know is whether we see any shift in, for example, in this case, the NDS values between the rearranged chromosome or scaffold and all the other scaffolds. So I plotted the distribution here, and you can see that you don't really see a shift. And so the reason why I was interested in that is because when you have rearrangements, and especially fusions, you can have changes in the linkage in that specific scaffold or chromosome, and that can definitely affect overall effects of selection, etc. But we couldn't really see anything, so we'll have to do some more studies later on this to further pursue this avenue. We also looked at key gene family evolution differences, and we found that on average, so Fasciculata has more multi-copy gene families than Labeldiana, and they are also on average larger, the multi-copy gene families in Fasciculata than in Labeldiana, which means that therefore there must be a higher rate in Fasciculata compared to Labeldiana. All of this is very interesting, but we are not really talking about how this actually relates to the different ecologies of these two plants. And so, as I said, we decided to focus on CAM photosynthesis, which they represent sort of the two extremes within the subgenus in phenotypic differentiation on this level. And we chose that for quite a few reasons, among others that CAM is actually a very evolvable trait. It has evolved about 35 times, in 35 plant families independently. And it is also of a lot of interest in general for agricultural purposes, etc. But another reason was that it's also a pretty well-described pathway. We sort of know what are the underlying genes, we know the enzymes, so that made it also perhaps a little bit more accessible to study. And I'll just briefly describe it. So a C3 plant, which is sort of the basic plant that does normal photosynthesis, if you want to see it that way, will perform its CO2 capture actually during the day. And that means that these leaf pores here, which is are the little holes through which the CO2 will come into the plant, they'll have to be open during the day, and that means that the plant can evaporate tons of water. CAM plants, on the other hand, they have moved the CO2 capture from the daytime to the nighttime. So they'll capture CO2 at night, but at night you can't actually make sugars because you don't have light. So instead of feeding it immediately into the chloroplast, the CO2 will be converted to malate and then accumulated in the vacuole overnight. And then during the day, again, it will be reconverted to CO2 and then converted to sugars. And so because there's a big temporal factor in here, and in fact, a lot of the underlying enzymes and genes already exist in a C3 plant, we decided to capture transcriptomic differences between the two species and actually sample them over a 24-hour cycle every 4 hours. So we had 6 time points, and we could call differentially expressed genes between the two species. So I'll just cut this short so that I have enough time for the other projects, but we then use these differentially expressed genes and also already pre-described smaller subset of pre-described CAM genes to link this to the other forms of variation we were seeing in between these two genomes. And the most salient result was actually that we found that the number of gene families that had differentially expressed genes or even CAM-related genes seem to be enriched for multi-copy gene families, specifically families that have a higher number in fasciculata than in Leiboldiana. So there's about 3 times more than in the normal genome. But also we saw an enrichment of multi-copy families that were not expanded. They have the same size, so they're probably more ancient duplications. And also on the functional level, so when we're not specifically looking at the genes, but at CAM-related functions, we also find that a lot of these functions tend to have higher gene copies in fasciculata than in Leiboldiana. So there seems to be an association here between those two, between the CAM C3 differences and gene family evolution. And we're wondering whether that might mean that when you have multi-copy gene families, you just have less restraint on the expression profiles of the genes, and they can evolve more freely. So we can conclude this project as that we actually picked up quite a few differences between the two species on many different levels: karyotype, rearrangements, TE landscape, etc. But when we look specifically at CAM differences, it's mostly found in the transcriptome and also in gene family evolution. And I think for me, the main takeaway message of this project was that historically, a lot of C3 CAM studies have been between very distantly related plants. For example, Arabidopsis and a monocot. We're talking about really big distances. And that can lead to overestimation of the number of changes that are actually needed to attain a certain trait. And if we look at more closely related pairs, we'll not overestimate this number of changes. We might, however, underestimate them. That is definitely a possibility. And so as we were working more and more on this CAM field and trying to understand CAM and reading the literature about it, it became more and more clear to me that while originally CAM and C3 were described as two very discrete traits where you had either a C3 plant or you had a CAM plant, just more and more reports have been coming in of intermediate forms of this phenotype. And there is kind of a shift that is happening at the time in this field of going from thinking about CAM photosynthesis as a discrete trait to more of a continuous trait. But it's complicated because a lot of people tend to just create new labels for certain types of intermediates rather than looking at it as a real continuum. And as we were looking into this, we were wondering more and more, well, okay, so what range of this full CAM continuum is the Tillandsia subgenus actually covering? Do we have, pure C3 plants that under absolutely have no pre-adaptation at all to CAM, or are we actually having CAM-enabled plants or C3 plants that when you stress them under drought, they will start to actually show some kind of a CAM cycle? And the same goes for our strong CAMs. Are they actually obligate CAMs or not? And this was something that we wanted to look into. So this project is more or less asking that, and also try to understand how the two extremes, let's say, react to drought, which is meant to be sort of the, well, in this model of an stress-induced evolutionary innovation, drought should kind of be the driver of this evolutionary trend. And a lot of this work was, by the way, done by Sarah. So I don't see her, but hey. So Sarah, thanks a lot for your contributions here, and let's look into what we found. So what we did was, again, select two extreme cases, let's say, in the subgenus. So again, we have Tillandsia leiboldiana, like in the previous study. For the CAM plant, we actually selected a stronger, even stronger CAM than fasciculata. They're very closely related, and the difference is not massive, but still, it's a different species. And the first thing that we did was to measure the acidity in the leaves of these plants at different time points. So as I said, a CAM plant would start to accumulate malate in the vacuole as a typical CAM cycle. And that will make the leaf more and more acidic. So if you measure the acidity, you can typically recover this CAM cycle. And that's what we did. And we did this for plants or leaves that were both in control conditions and also under drought conditions. So we didn't water them for 14 or 20 days. I think 14 days. And you can already see quite a I mean, there's a lot of interesting results here. So first of all, let's look at the strong CAM plant. It shows a very obvious depletion and then accumulation at night of malate, which is exactly what we are expecting for a CAM plant. But interestingly, at drought, it doesn't look very different. The only difference is perhaps at this time point a much faster accumulation of malate, but otherwise, also if you look at the delta, so this is the largest, the change in titratable acidity between the most extreme time points, they're very similar. For the C3 plant, however, in normal conditions, there isn't much of a trend. It varies a bit, but it's mostly flat, which is what a C3 plant, what we're also expecting for a C3 plant. But under drought, it does actually start to show a very weak CAM-like profile. It's nowhere near what the strong CAM plant is doing, but it's still doing something. So that was already something that we realized that, God, okay, maybe we're indeed not dealing with pure C3, but already an early form of weak CAM. And how does this look then on the transcription or the transcriptome level? So we called differentially expressed genes within species, always between day and night, at control and at drought. That's for these boxes here. And then the others are drought versus control differences at day and at night. And the first really obvious thing that pops out is that in drought conditions, our strong CAM plant is barely reacting. It has very few differentially expressed genes. And on the other hand, our weak CAM C3-like plant is actually doing a very strong reaction, especially at night. So that's a big difference, and I think it shows that they have very different phenotypes and different degrees of adaptation to drought. But then, yeah, the day and night differences are a bit more difficult to really see a pattern in because every organism will have constitutive day and night differences. But you can see that the CAM plant generally has a bit more, oh, sorry, I'm not sure if that was clear. So everything that is positive is upregulated at day. The negative counts are downregulated at day. And here it's the opposite. It's upregulated at drought, downregulated at drought. So in our weak CAM, we have mostly upregulation at day. That's a bit more equilibrated between the two time points in the CAM plant. And it seems to be shifting in both plants towards more upregulation in drought conditions at night. And then I was interested to know, where are the CAM genes, the ones that are already described as CAM genes by previous studies? Do they, are they shared between the two species? Are they, how much overlap is there in the CAM response between the two species and two phenotypes? And what I find, so this is the Venn diagram overlap of CAM genes specifically at day versus night. And I think what pops up is that a lot of the CAM genes are mostly actually uniquely expressed within one condition. And if we find sharing, it's actually mostly between conditions within a species, except for a couple of like more constitutional genes. But I do think that the response seems to be largely not shared. It's more difficult to say anything about sharedness in the drought versus control because we anyway have very few genes in the Infant But I did find it interesting that Tillandsia labeldiana at night has like 67 genes differentially expressed that are pre-described as CAM. So I do think that this shows that when labeldiana is stressing, it actually is activating a CAM-like cycle. So what can we take away from this part of the talk? Well, I think that what we tended to see as C3 Tillandsia are not actually pure Tillandsia. They have already a more early, they're like early forms of CAM, basically. And that means that when we study CAM evolution in the subgenus Tillandsia, we can't really say that we're studying the trait from 0 to 100, more like from 20 or 30 to 100. And that is perhaps a pity, because it would be nice to have the entire range, but I also think that it is probably quite expected between things that are very closely related, and it might be a reality for a lot of other plant lineages as well. And it still creates a lot of an interesting situation to study intermediate CAM phenotypes that we actually do not understand very well. The obligate CAM plant or strong CAM plant is clearly not reacting to the drought conditions much, so it seems to be much more adapted to it than the weak CAM. And it doesn't seem that the phenotypes are actually very similar. They seem to have different underlying genes. And when you looked at the physiological measurements, they also looked like the curves were quite different. And then for the last part of the talk, I hope I'm doing well on time. We're going to take a step back, as I said before as well, and stop looking at these key innovation traits. Instead, we're going to actually look at the radiation from a more phylogenetic framework and population genetics framework, and try to look into other potential processes. For example, what is the role? Do we find any demographic changes? Is that an important aspect of this radiation? Is gene flow an important aspect of this radiation? How pervasive is it? Do the key innovation traits actually act as a form of species barrier? Those are the questions we are trying to address in this project. And for that, we have a sampling of about 35 species that represent the whole, all the subclades of the subgenus Tillandsia. So we have the South American lineage here, and then the Central American lineage. I'm sure you can see that K21 is behaving a bit strangely. I'll talk about that later. And we also, it also involves a high diversity in terms of the key innovation traits as well. And this was sampled mostly by target capture sampling and a population level sampling. So usually trying to have as many individuals as we could. In some cases, we did end up with only 4 individuals per species, but that's what you do when you're in the field. So here, this is super preliminary, but I still want to show it. So it's results from demographic modeling that we're kind of doing at the moment. So we used approximate Bayesian computation to look into demographic change in the species. So these are very simple one population models where you basically say, well, is it expanding, contracting, or is the size being constant? And when did this more or less happen? How many generations ago? And so I'm plotting on this tree that represents a subset of the species I showed before. The South Americans are here, the Central Americans are here. How much this NE might have changed according to these models. So the bluer it is, the stronger the size seems to have increased, and if it's red, it's actually contracting. So we do see a bit of a difference between the two subclades. The Central American Tillandsias seem to be expanding and quite a bit, whereas the South Americans, we have a contracting species, and some of them are expanding, but they're not, the change is not nearly as strong. And I mean, this has to be taken with a grain of salt, of course, but when we use a generation time of 15 years, and we try to understand when this exactly happened, then we get to expansions that range between 3.5 to 1.7 million years ago, and this is kind of in the range of the origin of the clade as well. So it seems like it makes a little bit of sense in terms of the history that has also been discovered previously. And then we also wanted to understand gene flow, as I said. So we calculated these statistics across the clade, across the whole group. And so this heat map is showing you between species which ones might have had gene flow. So with these statistics, the more colorful it is, the more significant it is, basically. And you see very clearly that this clade K21 has a very distinct pattern where it seems to have had gene flow with the entire rest of the Central American group and also the South Americans. So we're thinking that we also have some additional phylonet results that also indicate this, that it's probably a hybrid clade, some form of a hybrid clade. And that will definitely be interesting to study further in the future. And even outside of that, we have pervasive gene flow in the other clades as well. So gene flow is definitely there. And so then we were wondering, can we see any link between the degree of gene flow between two species and whether they have the same key innervation trait or not? So what we did was to bin our D statistics into negative or positive values. So whenever we had a D statistic indicating gene flow between two species that have different ecologies, for example, one is a CAM and the other is a weak CAM plant, or one it has tank and the other one doesn't have tank, we would make it a negative D statistic. And whenever you had this between species that have the same key innervation trait, it would be positive. And then we can look at the distribution of the D statistics between negative and positive values and see if there is like a symmetric distribution on the two sides or if there is a shift towards positive values. And this is how it looks for the 6 key innervation traits that we tested. So you can see that some traits have a strong shift to positive values. So that seems to say that species with the same key innervation trait are more likely to have had gene flow. In some cases, that's not true. So for example, for the tank formation, we don't see such a case at all. And also for flower color, it's quite dubious if there really is an asymmetry. And there is one issue, though, with this analysis that we still need to address, which is that one thing is, of course, is this actually acting as a species, as a barrier for gene flow, or is it just that more closely related species are more likely to have the same key innervation trait and also more likely to have gene flow? And for that, we still need to come up with some correction on a phylogenetic level, which I'm very happy to hear suggestions if you have any. Okay, so with that, I would like to conclude my talk. So I think the 3 projects indicate that key innervation traits, especially CAM, photosynthesis, are relevant for this radiation and play an important role. We've seen that we have differences in how well adapted a species is to drought. We see this effect of gene flow and potential species barriers. But other processes seem to also have shaped this radiation and need a closer look, such as demographic history and also gene flow. And When it comes to CAM evolution, while we have quite a bit of genomic divergence between extreme cases of the subgenus, actually the changes that we find underlying CAM are rather small and more on the transcriptomic level, in the gene family level. And while Tillandsia does not seem to cover the full CAM, C3 to CAM spectrum, I think it's still a useful system to look into intermediate CAMs and further understand how we evolve from intermediate to obligate CAMs. So with that, I'd like to thank a lot of people who were involved in this. First of all, this is all thanks to Christian Lexer, who I had the chance to be a PhD student of in my first 3 months. And it's truly remarkable that in only 3 months, this man was able to convince me that Tillandsia is really fascinating. So I think that we can take that away from someone who was very enthusiastic and made me feel very welcome and is very missed. And then I also want to thank the people who stepped up, Ovidiu especially, who has been a huge guidance and help throughout the years, and Thibault, Carolina, Hannah Swardel, Joachim, Magnus, you for listening, and I'll take any questions.